So let's start the chapter 11, that is photosynthesis in higher plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all the other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis and are therefore called autotrophs. You have already learned that autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotrophs, and all other organisms that depend on green plants for food are called heterotrophs. Green plants carry out photosynthesis, a physiochemical process by which they use light energy to derive the synthesis of organic compounds. Ultimately, all living forms on earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on earth. Photosynthesis is important due to two reasons. It is the primary source of all food on earth. It also responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that transform light energy into chemical energy. Next is, what do we know? Let us try to find out what we already know about photosynthesis. Some simple experiment you may have done in the earlier classes have shown that chlorophyll, that is green pigment of the leaf, light and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black pepper, and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of light. Photosynthesis occurred only in the green parts of the leaves in the presence of light. Another experiment you may have carried out where a part of leaf is enclosed in a test tube containing some KOH soaked cotton which absorbs CO2 while the other half is exposed to air. The setup is then placed in light for some time. On testing for the presence of starch later in the two parts of the leaf, you must have found that the exposed part of the leaf tested positive for starch, while the portion that was in the tube tested negative. This showed that CO2 was required for photosynthesis. Can you explain how this conclusion could be drawn? ये जो सीरीज है पर चैप्टर की है, ऐसे मीन्स पूरी एनसीईआरटी का भी एक वन वीडियो में बना दूंगा, मीन्स ओनली इम्पोर्टेंट लाइंस वाली, पूरा चैप्टर की एक्सट्रैक्ट वाली मेरे पास ऑलरेडी है, हाईलाइटेड वाली. चलो तो नेक्स्ट, अर्ली एक्सपेरिमेंट्स. It is interesting to learn about those simple experiments that led to a gradual development in our understanding of photosynthesis. Joseph Priestley in 1733 to 1804 in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon get extinguished. Extinguished का मतलब क्या होता है पता है तुम्हें? बताओ क्या होता है? मतलब बताओ हिंदी में क्या होता है? मतलब पूजा हुआ, बूझना. Similarly, a mouse would soon suffocate in a closed space. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore to the air whatever breathing animal in the burning candle remove. ये important line है hypothesis की. Plant restore to the air whatever breathing animal and burning candle remove. Whatever breathing animal and burning candle removes. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight, Van Ingenhousz in 1730 to 1799 showed that sunlight is essential to the plant process that somehow purifies the air fouled by burning candles or breathing animals. Ingenhousz is in an elegant experiment with an aquatic plant showed that in bright sunlight, small bubbles were formed around the green part while in the dark they did not. Later he identified this bubble to be of oxygen, Hence he showed that it is the only green part of the plant that could release oxygen. Understood? It was not until about 1854 that Julius von Sachs provided evidence for production of glucose when plant grows. Julius von Sachs provided evidence for production of glucose when plant grows. Glucose is usually stored as starch. His later study showed that the green substance in plant chlorophyll as we know it now is located in special bodies later called chloroplast within the plant cells. He found that the green parts in plant is where glucose is made and that the glucose is usually stored as starch. ग्लूकोज कैसे स्टोर होता है बच्चों? स्टार्च की तरह. Now consider the interesting experiment done by T.W. Engelmann in 1843 to 1909. Using a prism, he split light into its spectral component and then illuminated a green alga, Cladopora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolution. He observed that the bacteria accumulated mainly in the line of blue and red light of the split spectrum. कौन से लाइन में? ब्लू एंड रेड लाइट, मतलब नीले और लाल कलर की लाइट में. A first action spectrum of the photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B. By the middle of this 19th century, the key feature of plant photosynthesis were known, namely that plant could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was then understood as CO2 plus H2O, and the presence of light gives CH2O plus O2. Whereas CH2O represented a carbohydrate, example glucose, as 6-carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by a microbiologist Cornelius Van Niel in 1897 to 1985. 1897 to 1985. वैसे सन इतने याद रखने के नहीं, आते भी नहीं है, कभी आया भी नहीं क्वेश्चन ईयर में से ऐसे. Who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen form a suitable oxidizable compound reduces carbon dioxide to carbohydrates. Based on his study of purple and green bacteria, photosynthesis is light dependent reaction in which hydrogen form a suitable oxidizable compound reduces carbon dioxide to carbohydrate. This can be expressed as 2H2A plus CO2 in the presence of light gives 2A plus CH2O plus H2O. In green plants, H2O is the hydrogen donor and is oxidized to oxygen and is oxidized to O2. Some organisms do not release oxygen during photosynthesis. Some organisms do not release oxygen during photosynthesis. When H2S instead is the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organism and not oxygen. Hence, he inferred that the oxygen evolved by the green plant comes from H2O. Hence, he inferred that the oxygen evolved by the green plant comes from H2O. Hence, he inferred that the oxygen evolved by the green plant comes from H2O, not from carbon dioxide. कहाँ से आती है? H2O से, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore 6CO2 plus 12H2O in the presence of light gives C6H12O6 plus 6H2O plus 6O2, where C6H12O6 represents glucose. नाम तो सुना ही होगा. The oxygen released is from water. The oxygen released is from water. कहाँ से रिलीज होती है? Water से. The oxygen released is from water. This was proved by using radioisotopic techniques. Radioisotope techniques. Note that this is not a single reaction, but description of multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given above? बताओ, करो explain. क्या explain करना है? Why 12 molecules of water as substrate are used in the equation given above? C6CO2 plus 12H2O. ये 12H2O क्यों किए याद है? Because for the production of one molecule of glucose, 12 NADPH molecules are required, and for production of 12 NADPH molecule, 12 water molecules are required. So this is the answer of that. Understood? So the next is, where does photosynthesis takes place? You would of course answer in the green leaf or in the chloroplast based on what you earlier read in chapter 8. You are definitely right. Photosynthesis does take place in the green leaves of the plant, but it does so also in other green parts of the plants. Can you name some other parts where you think photosynthesis may occur? That are in stems, petioles, roots, flower, fruits and seeds. Yes, photosynthesis can occur in other parts of the plant in addition to leaves, including stems, petioles, roots, flower, fruit and seeds. Photosynthesis can occur in any part of the plant, that is chlorophyll. You would recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually, the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplast will be aligned with their flattened surface parallel to the wall? When would they be the perpendicular to the incident light? Answer me. Socho. So, the chloroplast in plant cell align with their flat surface parallel to the walls of the mesophyll cells when the light intensity is low or optimum. जब लो और ऑप्टिमम होगी तो पैरेलल, and the chloroplast align perpendicular to the incident line when the light intensity is very high. Understood? So the next is, you have studied the structure of the chloroplast in chapter 8. Within the chloroplast, there is a membranous system consisting of grana, the stroma lamellae, and the matrix stroma. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reaction synthesize sugar, which in turn form a starch. The former set of reactions, since they are directly light dependent, are driven and are called light reaction, photochemical reactions. The latter are not directly light driven but are dependent on the product of light reactions ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reactions, carbon reactions, or light reaction, photochemical reaction, and dark reaction are carbon reactions. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. So the next is, how many types of pigment are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves? Even in the same plant, we can look for an answer to this question by trying to separate the leaf pigment of any green plant through paper chromatography. A chromatography separation of the leaf pigment shows that the color that we see in leaves is not due to a single pigment, but due to 4 pigments: chlorophyll A, bright or blue green, and the chromatograph. कौन सा कलर है? क्लोरोफिल ए का ब्राइट और ब्लू ग्रीन, और क्लोरोफिल बी का है येलो ग्रीन, क्लोरोफिल बी का है येलो ग्रीन, बेलो ग्रीन मीन्स बी का येलो ग्रीन, सैंथोफाइल्स का है येलो, एंड कार्टिनोइड्स का है येलो टू येलो ऑरेंज, कार्टिनोइड्स का है येलो टू येलो ऑरेंज, और सैंथोफाइल का येलो. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at a specific wavelength. Can you guess which is the most abundant plant pigment in the world? कौन सा plant pigment? The answer is chlorophyll. Let us study the graph showing the ability of chlorophyll A pigment to absorb light of different wavelength. Of course, you are familiar with the wavelength of the visible spectrum of light as well as the visual. Can you determine the wavelength color of light at which chlorophyll A shows the maximum absorption from the figure 11.3a? Check the figure in the book. Does it show another absorption peak at any other wavelengths too? If yes, then which one? Now look at figure 11.3b showing the wavelength at which maximum photosynthesis occurs in a plant. Can you see that the wavelength at which there is a maximum absorption by chlorophyll A, that is in the blue and the red line, maximum absorption is there. also show higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at the figure 11.3c, can you say that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? These graphs together show that most of the photosynthesis takes place in the blue and red line of the spectrum. In the blue and red line of the spectrum, most of the photosynthesis takes place. Some photosynthesis does take place at the other wavelength of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigment like chlorophyll B, xanthophylls and catenoids, which are called accessory pigment, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. Indeed, they not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from the photooxidation. So the next is, what is a light reaction? Light reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediate ATP and NADPH, that is ATP and NADPH, high energy chemical intermediate. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes LHC within the photosystem 1, that is PS1, and photosystem 2, that is PS2. These are named in the sequence of their discovery. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The light harvesting complex are made up of hundreds of pigment molecules bound to protein. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system, also called as antennae. This pigment help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll second A molecule forms the reaction center. The single chlorophyll second A molecule forms the reaction center. The reaction center is different in both the photosystems. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, understood, and it is called P700, while in the PS2 it is an absorption maxima at 680 nanometer and is called P680. So the reaction center of the chlorophyll A in PS1, it's 700 nanometer and is called P700, and PS2 का है P680. ठीक है, absorption maxima at 680 nanometer. मैक्सिमा की जगह मिनिमा कर देंगे, बट करेक्ट है मैक्सिमम, अंडरस्टूड. नेक्स्ट इज द इलेक्ट्रॉन ट्रांसपोर्ट. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electron to become excited and jump into orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor, which passes them to an electron transport system consisting of cytochromes. This movement of electron is downhill in terms of oxidation, reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of the photosystem PS1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again this time to a molecule of energy-rich NADP+. The addition of these electrons reduces NADP plus to NADPH plus H. The addition of this electron reduces NADP plus to NADPH plus H plus. This whole scheme of transfer of electron starting from the PS second uphill to the acceptor down the electron transport chain to PS1 excitation of electron transfer to another acceptor and finally downhill to NADP plus reducing it to NADPH plus H plus. H plus है हाइड्रोजन के ऊपर वाला प्लस है और वो वाला है NADPH के बीच वाला प्लस is called the Z scheme due to its characteristic shape. This shape is formed when all the carriers are placed in a sequence on a redox potential scale. Next is splitting of water. You would then ask, how does PS second supply electron continuously? The electron that were moved from photosystem second must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the PS second. Water is split into 2H positive, oxygen and electrons. Is associated. The splitting of water is associated with PS2. Water is split into 2H positive, oxygen and electron. किस में होता है वाटर स्प्लिट 2H पॉजिटिव में, ऑक्सीजन में, इलेक्ट्रॉन्स में. This creates oxygen, one of the Neet product of photosynthesis. The electron needed to replace those removed from the photosystem 1 are provided by photosystem 2. The reaction given here is 2H2O gives 4H positive plus oxygen plus 4 electron. We need to emphasize here that the water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Then why are the proton and oxygen formed likely to be released in the lumen or on the outer side of the membrane? Answer: where are the protons and oxygen formed likely to be released in the lumen or on the outer side of the membrane? कहाँ पे? Proton are released in the lumen of the thylakoid, while oxygen is released outside the thylakoid membrane. Understood? So the next is cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substance and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystem works in a series, first PS second and then the PS first, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH plus H positive are synthesized by this kind of electron flow. H positive का मतलब है H के ऊपर वाला प्लस, ठीक है. Both ATP and NADPH plus H positive are synthesized by this kind of electron flow. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electron. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electron. A possible location where this could be happening is in the stroma lamellae. While the membrane or lamellae of the grana have both PS1 and PS2, the stroma lamellae membrane lacks PS2 as well as NADP reductase enzyme. स्ट्रोमा लेमेले में क्या क्या नहीं होता है, PS2 नहीं होता है और NADP reductase enzyme नहीं होता है. The excited electron does not pass on to the NADP positive but is cycled back to the PS1 complex through the electron transport chain. The cyclic flow hence results only in synthesis of ATP but not of NADPH plus H positive. Cyclic phosphorylation also occurs when only light of wavelength beyond 680 nanometer are available for excitation. कब होगा cyclic photophosphorilation? जब light of wavelength होगी beyond 680 nanometer. जब light होगी beyond 680 nanometer are available for excitation. Means 680 nanometer से आगे की होगी. Next is chemi-osmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemi-osmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to the development of a proton gradient across a membrane. This time, these are the membranes of thylakoid. There is one difference too. Here, the proton accumulation is towards the inside of the membrane, that is, in the lumen. In respiration, proton accumulate in the intermembrane space of the mitochondria. When electron moves through the ETS, in respiration, proton accumulate in the intermembrane space of the mitochondria when electrons move through the ETS, in electron transport system. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that take place during the activation of electrons and their transport to determine the steps that cause a proton gradient to develop. Point A. Since splitting of the water molecule takes place on the inner side of the membrane, the proton or hydrogen ion that are produced by the splitting of water accumulate within the lumen of thylakoids. Splitting of water molecule inner side में होती है membrane की, तो proton कहाँ पे accumulate होंगे? Proton और hydrogen ion that are produced accumulate within the lumen of the thylakoid. Understood? So the next point is B. As electron move through the photosystem, protons are transported across the membrane. This happens because the primary acceptor of the electron, which is located towards the outer side of the membrane, transfer its electron not to an electron carrier but to an hydrogen carrier. Not to an electron carrier but to the hydrogen carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. Into the inner side or the lumen side of the membrane. Point C. The NADP reductase enzyme is located on the stroma side of the membrane. कहाँ पे located होता है NADP reductase enzyme? Stroma की side पे of the membrane. Along with the electrons that come from the acceptor of the electrons of PS first protons are necessary for the reduction of NADP positive to NADPH plus H positive. पॉजिटिव का मतलब H के ऊपर वाला पॉजिटिव मीन्स प्लस, उसको पॉजिटिव बोल रहा हूँ मैं यहाँ पे. These protons are also removed from the stroma. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number. Proton in the stroma decrease in number, while in the lumen there is an accumulation of protons. स्ट्रोमा में क्या होंगे, डिक्रीज होंगे और ल्यूमन में एक्यूमुलेशन होगा. दिस क्रिएट्स ए प्रोटॉन ग्रेडियंट अक्रॉस द थाइलाकोइड मेम्ब्रेन as well as measurable decrease in pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of the proton across the membrane to the stroma through the transmembrane channel of the CF node of the ATP synthesis. The gradient is broken down due to the movement of proton across the membrane to the stroma through the transmembrane channels of the CF node of the ATP synthesis. ये डबल बार लाइन्स इसलिए पढ़ रहा हूँ ताकि माइंड में उतर जाए, इसलिए मैं स्लोअर जहाँ पे इम्पोर्टेंट है वहाँ पे डबल बार रिपीट कर रहा हूँ. द ATP synthesis enzyme consists of two parts. One called the CF node is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthesis, which makes the enzyme synthesize several molecules of energy-packed ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient and ATP synthesis. यहाँ से बहुत बार क्वेश्चन आया. Chemiosmosis requires a membrane, a proton pump, a proton gradient and ATP synthesis. Energy is used to pump proton across a membrane to create a gradient or a high concentration of proton within the thylakoid lumen. ATP synthesis has a channel that allows diffusion of protons back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. This releases enough energy to activate ATP synthesis enzyme that catalyzes the formation of ATP. ATP synthase has a channel that allows diffusion of proton back across the membrane. This releases enough energy to activate ATP synthesis enzyme that catalyzes the formation of ATP. Along with the NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugars. Next is, where are the ATP and NADPH used? We learnt that the products of the light reaction are ATP, NADPH and O2, that is oxygen. Of this, oxygen diffuses out of the chloroplast, while ATP and NADPH are used to drive the processes leading to the synthesis of food, more accurately sugars. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light, but is dependent on the product of the light reaction, that is ATP and NADPH. Besides CO2 and H2O, you may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss this amongst yourself. करो discuss, ठीक है? Let us now see how the ATP and NADPH are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2O ब्रैकेट क्लॉज N, CH2O ब्रैकेट में है और उसके बाहर N और sugars. It was of interest to scientists to find out how this reaction proceeded, or rather what was the first product formed when CO2 is taken into a reaction or fixed. Just after World War II, among the several efforts to put radioisotopes to beneficial use, the work of Melvin Calvin is exemplary. The use of radioactive 14-carbon by him in algal photosynthesis studies led to the discovery that the first CO2 fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway, hence it is called Kelvin cycle after him. केल्विन जी छोड़ गए उनके पीछे केल्विन साइकिल जो हम पढ़ रहे हैं। अरे केल्विन जी। द फर्स्ट प्रोडक्ट आइडेंटिफाइड वाज 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also tried to know whether all plants have PGA as the first product of CO2 fixation, or whether any other product was formed in other plants. Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of CO2 fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, or OAA. Since then, CO2 assimilation during photosynthesis was said to be of 2 main types. Those plants in which the first product of CO2 fixation is a C3 acid PGA, that is the C3 pathway, and those in which the first product was C4 acid OAA, oxaloacetic acid, that is the C4 pathway. These two group of plants showed other associated characteristics that we will discuss later. So the next is the primary acceptor of CO2. Let us now ask ourselves a question that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which, after accepting fixing CO2, would have 3 carbons of PGA? पागल के क्वेश्चन कर रखे हैं. The studies were unexpectedly showed that the acceptor molecule was a 5-carbon keto sugar ribulose 5-phosphate RUBP. Did any of you think of this possibility? Do not worry, the scientists also took a long time and conducted many experiments to reach this conclusion. They also believed that since the first product was a C3 acid, the primary acceptor would be a 2-carbon compound. They spent many years trying to identify a 2-carbon compound before they discovered the 5-carbon RUBP. The study were unexpectedly showed that the acceptor molecule was a 5-carbon keto sugar ribulose bisphosphate. RUBP और 5-carbon की keto sugar है, ये important है बस, ठीक है. The next is the Kelvin cycle. आओ पधारो जी Kelvin जी आए हैं. Kelvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. the RUBP was regenerated. That the pathway operated in a cyclic manner, the RUP was regenerated. Let us now see how the Calvin pathways operates and where the sugar is synthesized. Let us at the outset understand very clearly that the Calvin pathway occur in all photosynthetic plants. In all photosynthetic plants, it does not matter whether they have C3 or C4 or any other pathways. It occurs in all photosynthetic plants. For ease of understanding, the Calvin cycle can be described under three stages: carboxylation, reduction, and regeneration. Carboxylation is the first. Carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of the Calvin cycle, where CO2 is utilized for the carboxylation of RUBP. Most crucial step है carboxylation. This reaction is catalyzed by the enzyme RUBP carboxylase. कौन सा एंजाइम से? RUBP carboxylase. लिख डालो माइंड में, RUBP carboxylase, which result in the formation of two molecules of 3-PGA. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase oxygenase or Rubisco. Rubisco का फुल नेम क्या है? RUBP carboxylase oxygenase. यह भी कह सकते हैं Rubisco को, ठीक है. The Calvin cycle proceeds in the three-stage carboxylation, during which CO2 combine with ribulose 1,5-bisphosphate. Second is reduction, during which carbohydrate is formed at the expense of the photochemically made ATP and NADPH. And third is the regeneration, during which the CO2 acceptor ribulose 1,5-bisphosphate is formed again, so that the cycle continues. ये जो फिगर के नीचे डिटेल थी वहाँ से पढ़ा है. अब अपन पढ़ रहे हैं सेकंड पॉइंट, रिडक्शन. These are series of reaction that lead to the formation of glucose. The step involves utilization of molecules of ATP for phosphorylation. The step involves utilization of two molecules of ATP for phosphorylation and 2 of NADPH for reduction per CO2 molecule fixed. The fixation of 6 molecules of CO2 and 6 turns of the cycle are required for the formation of 1 glucose molecule from the pathway. For 1 molecule of glucose from the pathway, कितनी? Fixation of 6 molecule of CO2 and 6 turns of cycle are required for 1 molecule of glucose. Third is regeneration. Regeneration of the CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP. The regeneration steps require 1 ATP for phosphorylation to form RUBP. The regeneration steps require 1 ATP for phosphorylation to form RUBP. Important line है, डबल पढ़ता हूँ. The regeneration steps require 1 ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 of NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of the cycle are required. Work out how many ATP and NADPH molecules will be required to make 1 molecule of glucose through the Calvin pathway. क्वेश्चन क्या पूछा है? Work out how many ATP and NADPH molecules will be required to make 1 molecule of glucose through the Calvin pathway. तो ATP होगी 18 और NADPH होंगे 12. ठीक है. It might help you to understand all of this if we look at what goes in and what comes out of the Calvin cycle. In अंदर जाती हैं 6 CO2, out बाहर आती हैं 1 ग्लूकोज. इन होती है 18 ATP, आउट आती है 18 ADP. ATP गई ADP आई. वाह. इन होते हैं 12 NADPH, आउट आते हैं 12 NADP. एच खा जाता है. जाते हैं अंदर 12 NADPH, आते हैं बाहर NADP 12 NADP. एच को छोड़ के आ जाते हैं. The C4 pathway. Plants that are adapted to dry tropical rain have the C4 pathway mentioned earlier. Important topic है, यहाँ से क्वेश्चन आते ही आता है. Two, these plants have the C4 oxaloacetic acid as the first CO2 fixation product. They use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. They use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then in what way are they different from C3 plants? This is a question that you may reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate high temperatures. They show a response to high light intensity. They lack a process called photorespiration and have greater productivity of biomass. C4 plant are special. They have a special type of leaf anatomy. They tolerate high temperature. They show a response to high light intensity. They lack a process called photorespiration and have greater productivity of biomass. Let us understand this one by one. Study vertical section of leaves, one of a C3 plant and the other of a C4 plant. Do you notice the differences? Do both have the same type of mesophiles? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundles of the C4 plant are called bundle sheath cells. The particularly large cells around the vascular bundles of the C4 plants are called bundle sheath cells, and the leaves which have such anatomy are said to have crange anatomy. Crange means wreath and is a reflection of the arrangement of cells. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by having a large number of chloroplasts, thick wall, impervious to gaseous exchange, and no intercellular spaces. You may like to cut a section of the leaves of C4 plants, example are maize or sorghum, to observe the crange anatomy and the distribution of mesophile cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical section of the leaves. Observe under the microscope, look for the bundle sheath around the vascular bundles. The presence of the bundle sheath would help you to identify the C4 plants. Now study the pathway shown in figure 11.9. देख लेना, ठीक है पेज. This pathway that has been named the H and slack pathway is again a cyclic process. Let us study the pathway by listing the steps. This pathway that has been named the H and slack pathway is again a cyclic process. फिगर देख सकते हो आप वीडियो में, ठीक है. Let us study the pathway by listing the steps. The primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate, phosphoenol pyruvate, PEP, and is present in the mesophyll cells. The enzyme responsible for the fixation is PEP carboxylase or PEPcase. It is important to register that the mesophyll cells lacks Rubisco enzyme. The C4 acid OAA oxaloacetic acid is formed in the mesophyll cells. It then forms other 4-carbon compound like malic acid or aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acid are broken down to release CO2 and a 3-carbon molecule. ये paragraph important है. I am repeating it again. It then form other 4-carbon compound like malic acid or aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acid are broken down to release CO2 and a 3-carbon molecule. The 3-carbon molecule is transported back to the mesophyll where it is converted to PEP again, thus completing the cycle. The 3-carbon molecule is transported back to the mesophyll cell where it is converted to PEP again, thus completing the cycle. The CO2 released in the bundle sheath cells enter the C3 or the Calvin pathway. The CO2 released in the bundle sheath cells enters the C3 or the Calvin pathway, a pathway common to all plants. The bundle sheath cells are rich in enzyme ribulose bisphosphate carboxylase oxygenase, that is Rubisco, but lack PEPcase. बंडल शीट में Rubisco होता है, ठीक है, बहुत ज़्यादा रिच, और लेक होता है, मतलब होता नहीं PEPcase, लेक. Thus the basic pathway that results in the formation of the sugars, the Calvin pathway, is common to the C3 and C4 plant. Did you note that the Calvin pathway occurs in all mesophyll cells of the C3 plant? In the C4 plant, it does not take place in the mesophyll cell, but it does so only in the bundle sheath cells. In the C4 plant, it does not take place in the mesophyll cells, but does so only in the bundle sheath cells. Did you note that the Calvin pathway occurs in all the mesophyll cells of the C3 plant? In the C4 plant, it does not take place in the mesophyll cell, but does so only in the bundle sheath cells. Understood? डायग्राम पढ़ देता हूँ एक बार, ठीक है, माइंड में चला लेना. ऊपर है मेसोफाइल सेल है, नीचे बंडल शीट सेल है. स्टार्ट हुआ एटमॉस्फेरिक CO2 से अंदर गया HCO3 फॉस्फोइनॉल पायरुवेट का फिक्सेशन हुआ C4 एसिड, ट्रांसपोर्ट हुआ बंडल शीट सेल में C4 एसिड प्लाज्मोडस मेटा के थ्रू, डिकार्बोक्सिलेशन हुआ, वहाँ से CO2 निकली फिक्सेशन बाय केल्विन साइकिल, और C3 एसिड निकला जो ट्रांसपोर्ट होके बंडल शीट सेल से वापस कहा गया मेसोफाइल सेल में. वहाँ पे C3 एसिड और C3 एसिड से रीजनरेशन हुआ फॉस्फोइनॉल पायरुवेट. जब मेसोफाइल सेल दे रखा है, उसकी प्लाज्मा मेम्ब्रेन दे रखी है और सेल वॉल दे रखी. पूरा डायग्राम भी पढ़ दिया है मैंने, ठीक है. कौन सा वाला डायग्राम? 11.9 वाला डायग्रामेटिक रिप्रेजेंटेशन ऑफ हैच एंड स्लैग पैथ. नेक्स्ट इज फोटो रेस्पिरेशन. Let us try and understand one more process that creates an important difference between C3 and C4 plant. That is photorespiration process. कौन सी process? Photorespiration process. To understand photorespiration process, we have to know a little bit more about the first step of the Calvin pathway. The first CO2 fixation step, this is the reaction where RUBP combines with CO2 to form two molecules of 3-PGA. That is catalyzed by Rubisco. RUBP plus CO2 in the presence of Rubisco gives 2 into 3-PGA. Rubisco, that is the most abundant enzyme in the world. Do you wonder why? Is characterized by the fact that it is active site can bind to both CO2 and O2. Hence the name. Rubisco, that is the most abundant enzyme in the world, is characterized by the fact that its active site can bind to both CO2 and O2. Hence the name. Can you think how this could be possible? Rubisco has a much greater affinity for CO2. When the CO2 is to O2 is nearly equal. जब CO2 और O2 का रेशियो इक्वल होगा तो ग्रेटर एफिनिटी होगी CO2 के लिए. Imagine what would happen if this were not so. This binding is competitive. It is the relative concentration of oxygen and CO2 that determines which of the two will bind to the enzyme. In C3 plants, some oxygen does bind to Rubisco and hence CO2 fixation is decreased. Here, the RUBP instead of being converted to two molecules of PGA binds with the oxygen to form one molecule of phosphoglycerolate and phosphoglycolate, 2 carbon, in a pathway called photorespiration. In the photorespiratory pathway, there is neither synthesis of sugar nor of ATP. ना तो ATP बन रही है ना sugar बन रही है. Rather, it results in the release of CO2 with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not yet known. The biological function of photorespiration is not known yet. In C4 plants, the photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. In C4 plant, photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. यहाँ से assertion reason बन सकता है या फिर statement भी बन सकता है. ठीक है. This takes place when the C4 acid from the mesophile is broken down in the bundle sheath cell to release CO2. This results in increasing the intracellular concentration of CO2. In turn, this ensures that the Rubisco functions as a carboxylase, minimizing the oxygenase activity. Carboxylase minimizing the oxygenase activity. फंक्शन कर रहा है कार्बोक्सिलेज की तरह. किसकी एक्टिविटी को मिनिमाइज करके? ऑक्सीजनेस. नाउ दैट यू नो दैट द C4 प्लांट lack photorespiration. Now that you know that the C4 plant lack photorespiration. क्या लैक करते हैं? Photorespiration. You probably can understand why productivity and yields are better in these plants. In addition, these plants show tolerance to higher temperature. Based on the above discussion, can you compare plants showing C3 and C4 pathway? Use the table given in the table 11.1 and fill in the information. कर सकते हो information? पढ़ोगे? क्यों? पढ़ना है क्या? चलो आ जाओ देन. टेबल पढ़ देता हूँ विद आंसर्स के साथ. बहुत इंपॉर्टेंट टेबल है. यहाँ से बहुत क्वेश्चंस डायरेक्ट आपके माइंड में सेट हो जाएंगे. ठीक है. चलो. कैरेक्टरिस्टिक्स. फर्स्ट, सेल टाइप इन विच द केल्विन साइकिल टेक्स प्लेस. C3 प्लांट्स में मीजोफाइल सेल्स में होती है केल्विन साइकिल और C4 प्लांट्स में बंडल शीथ सेल में होती है. ठीक है. कैरेक्टरिस्टिक, सेल टाइप इन विच द इनिशियल कार्बोक्सिलेशन रिएक्शन occurs. C3 प्लांट्स में मीसोफाइल में होती है और C4 प्लांट्स में भी मीसोफाइल सेल्स में होती है. हाउ मेनी सेल टाइप डज द लीफ हैव दैट फिक्स CO2? C3 प्लांट में 1 होते हैं मीसोफाइल और C4 प्लांट में 2 होते हैं बंडल शीथ सेल्स और मीसोफाइल सेल्स. अंडरस्टूड? ठीक है. आगे समझना. So the next is, what is the primary CO2 acceptor? C3 प्लांट में है RUBP राइबुलोज बिसफॉस्फेट, और C4 प्लांट में है PEP फॉस्फोइनोल पाइरुवेट। नंबर ऑफ कार्बन्स इन द प्राइमरी CO2 एक्सेप्टर, C3 प्लांट्स में होते हैं 5 कार्बन, 5 नंबर ऑफ कार्बन इन द प्राइमरी CO2 एक्सेप्टर C3 प्लांट में, और 3 होते हैं C4 प्लांट में। नंबर ऑफ कार्बन इन द प्राइमरी CO2 एक्सेप्टर जो होता है उसमें कितने कार्बन होते हैं, तो C4 प्लांट में 3 होते हैं, C3 में 5 होते हैं। अंडरस्टूड? ठीक है। विच इज़ द प्राइमरी CO2 फिक्सेशन प्रोडक्ट, C3 प्लांट में होता है PGA और C4 प्लांट में होता है OAA ऑक्सलो एसिटिक एसिड, और C3 में होता है फॉस्फोग्लाइसरिक एसिड। नंबर ऑफ कार्बन इन द प्राइमरी CO2 फिक्सेशन प्रोडक्ट में C3 प्लांट्स में होंगे 3 और C4 प्लांट्स में होंगे 4। डज द प्लांट हैव रूबिस्को, C3 में भी यस और C4 प्लांट में भी यस, दोनों में होगा। डज द प्लांट हैव पैपकेस, C3 प्लांट में पैपकेस नहीं होता है और C4 प्लांट में पैपकेस होता है। ठीक है। विच सेल्स इन द प्लांट हैव रूबिस्को, C3 प्लांट में मीजोफाइल में होता है, C4 प्लांट में बंडल शीट में होता है। CO2 फिक्सेशन रेट अंडर हाई लाइट इंटेंसिटी, C3 प्लांट में मीडियम होती है और C4 प्लांट में हाई, क्योंकि हाई लाइट इंटेंसिटी पे वर्क करते हैं। वेदर फोटो रेस्पिरेशन इज प्रेजेंट एट लो लाइट इंटेंसिटी, C3 प्लांट्स में हाई में होता है और C4 प्लांट में नेग्लिजिबल होता है। एट लो लाइट इंटेंसिटी पे हाई होता है। ठीक है, फोटो रेस्पिरेशन C3 प्लांट्स में। वेदर फोटो रेस्पिरेशन इज प्रेजेंट एट हाई लाइट इंटेंसिटी में भी हाई होता है C3 प्लांट में, और C4 प्लांट्स में तो होता ही नहीं, नेग्लिजिबल। वेदर फोटो रेस्पिरेशन वुड बी प्रेजेंट एट लो CO2 कंसेंट्रेशन, सब में हाई होता है C3 में, बाकी नेग्लिजिबल होता है C4 में, होता ही नहीं फोटो रेस्पिरेशन। वेदर फोटो रेस्पिरेशन वुड बी प्रेजेंट एट हाई CO2 कंसेंट्रेशन, हाई होता है C3 प्लांट्स में और नेग्लिजिबल होता है C4 प्लांट्स में। टेम्परेचर ऑप्टिमम क्या है C3 प्लांट के लिए, 20 से 25 डिग्री सेल्सियस, ये बहुत इंपॉर्टेंट है। और C4 के लिए होता है 30 से 40 डिग्री सेल्सियस। जो ऑप्टिमम टेम्परेचर है C3 प्लांट के लिए 20 से 25 डिग्री सेल्सियस है, और C4 के लिए 30 से 40 डिग्री सेल्सियस है। एग्जाम्पल जो है C3 के वीट और राइस है, C4 के मैज और शुगर केन। ठीक है, मैज और शुगर केन। टेबल पूरी दिमाग में उतार दी। ठीक है। नेक्स्ट इज फैक्टर अफेक्टिंग फोटोसिंथेसिस। An understanding of the factor that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. Photosynthesis is under the influence of several factors, both internal and external. The plant factor include the number, size, age, and orientation of leaves, mesophyll cells, and chloroplast. Internal CO2 concentration and the amount of chlorophyll. प्लांट फैक्टर कौन से हैं? नंबर, साइज, एज एंड ओरिएंटेशन ऑफ लीव, मेसोफाइल सेल्स एंड क्लोरोप्लास्ट, इंटरनल CO2 concentration and amount of chlorophyll. The plant or the internal factors are dependent on the genetic predisposition and the growth of the plant. आंखें बंद करके सुनते जाओ बस, जो भी सारे पॉडकास्ट है। ठीक है। द एक्सटर्नल फैक्टर्स वुड इंक्लूड द अवेलेबिलिटी ऑफ द सनलाइट, टेम्परेचर, CO2 concentration and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Internal CO2 concentration किसका फैक्टर है? प्लांट का फैक्टर है। ठीक है। और CO2 concentration है एक्सटर्नल फैक्टर। As a plant photosynthesis, all these factors will simultaneously affect its rates. Hence, though several factors interact with and simultaneously affect photosynthesis or CO2 fixation. Usually, one factor is the major cause or is the one that limits the other. Hence, at any point, the rate will be determined by the factor available at suboptimal level. Rate will be determined by the factor available at suboptimal levels. When several factors affect any biochemical processes, Blackman's in 1905 law of limiting factor comes into effect. This states the following: if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. It is the factor which directly affects the process if its quantity is changed. If a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. For example, despite the presence of a green leaf and optimal light and CO2 condition, the plant may not photosynthesize if the temperature is very low. This leaf, if given the optimal temperature, will start photosynthesizing. Next is factor light. We need to distinguish between the light quality, light intensity, and the duration of exposure to light. While discussing light as a factor that affects photosynthesis, there is a linear relationship between incident light and CO2 fixation rate at low light intensity. At higher light intensity, gradually the rate does not show further increase as other factors become limiting. What is the interesting to note is that light saturation occurs at 10% of the full sunlight. At 10% of the full sunlight, ये important है. फुल की जगह कुछ भी कर सकते हैं चेंज करके हाफ या कुछ, बट फुल करेक्ट है. Hence, except for plants in shade or in dense forest, light is rarely a limiting factor in nature. Hence, except आ रहा है यहाँ पे, except for plants in shade or in dense forest, light is rarely a limiting factor in nature. Increase in incident light beyond a point causes the breakdown of chlorophyll and a decrease in photosynthesis. जब एक पॉइंट से ऊपर चल जाएगी इंक्रीज इन इंसिडेंट लाइट, तो वो क्या करेगी, ब्रेक डाउन करेगी क्लोरोफिल को और डिक्रीज करेगी फोटोसिंथेसिस को. नेक्स्ट फैक्टर इज कार्बन डाइऑक्साइड कंसंट्रेशन. कार्बन डाइऑक्साइड इज द मेजर लिमिटिंग फैक्टर for photosynthesis, major limiting factor of the photosynthesis. The concentration of CO2 is very low in the atmosphere, between 0.03 and 0.04%. Increase in concentration up to 0.05% can cause an increase in CO2 fixation rates. Beyond this, the level can become damaging over longer periods. The C3 and C4 plants respond differently in CO2 concentration. At low light conditions, neither group responds to high CO2 conditions. At a high light intensities, both C3 and C4 plants show increase in the rate of photosynthesis. What is important to note is that the C4 plant shows saturation at about 360 mu L inverse minus 1, ठीक है. While C3 responds to increased CO2 concentration and saturation is seen only beyond 450 mu L inverse means minus 1 micro U वाला, ठीक है. इसको ऐसे रिमेंबर कर सकते हैं, C4 के बाद 360 और C3 के बाद 450. 3 के बाद 4 आता है तो 450, ठीक है. 4 के बाद आता है 3, ठीक है, उल्टा है, ठीक है. C4 show saturation about 360 while C3 show 450. Understood? 4 में 3 के बाद 4. Thus, current availability of CO2 levels is limiting to the C3 plants. C3 का saturation point है beyond 450 और C4 का है 360, ठीक है. अब माइंड में उतर जाना चाहिए. The fact that C3 plant respond to higher CO2 concentration by showing increased rates of photosynthesis leading to higher productivity has been used for some greenhouse crops such as tomato and bell pepper. The fact that C3 plant respond to higher CO2 concentration by showing increased rate of photosynthesis leading to higher productivity have been used for some greenhouse crops such as tomato and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads to the higher yield. Understood? So the next is temperature. The dark reaction being enzymatic are temperature controlled. Though the light reaction are also temperature sensitive, they are affected to a much lesser extent. Though the light reaction are also temperature sensitive, they are affected to a much lesser extent. The C4 plant respond to higher temperature and show higher rate of photosynthesis, while C3 plant have a much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depend on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plant adapted to temperate climates. Next is water. Even though water is one of the reactants in the light reaction, the effect of water as factor is more through its effect on the plant rather than directly on photosynthesis. Even though water is one of the reactants in the light reaction, the effect of water as a factor is more through its effect on the plant rather than directly on photosynthesis. Is more through its effect on the plant rather than directly on photosynthesis. किस पे कर रहा है इफेक्ट प्लांट पे फोटोसिंथेसिस की वजह. Water stress causes the stomata to close, hence reducing the CO2 availability. Besides, water stress also makes leaves wilt, thus reducing the surface area of leaves and their metabolic activity as well. So the summary we are going to read in a speedy way. Green plants make their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in by the leaves through the stomata and used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in the green parts of the plant, mainly the leaves. Within the leaves, the mesophyll cells have a large number of chloroplasts that are responsible for CO2 fixation. Within the chloroplast, the membranes are sites for the light reaction, while the chemosynthetic pathway occurs in the stroma. Photosynthesis has two stages, the light reaction and the carbon fixing reaction. In the light reaction, the light energy is absorbed by the pigment present in the antenna and funneled to a special chlorophyll A molecule called a reaction center, chlorophylls. There are two photosystems, PS1 and PS2. PS1 has a 700 nanometer absorbing chlorophyll A P700 molecule at its reaction center, while PS2 has a P680 reaction center that absorbs red light at 680 nanometer. After absorption, light electrons are excited and transferred through PS2 and PS1 and finally to NAD forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the proton gradient due to the movement through the F0 part of the ATPase enzyme releases enough energy for synthesis of ATP. Splitting of water molecule is associated with the PS2, resulting in the release of O2, proton, and transfer of electron to PS2. Splitting of water is associated with PS2, resulting in release of oxygen, proton, and transfer of electron to PS2. In the carbon fixation cycle, CO2 is added by the enzyme Rubisco to a 5-carbon compound RUBP that is converted to 2 molecules of 3 carbon PGA. This is then converted to sugar by the Calvin cycle and the RUBP is regenerated. During this process, ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful oxygenation reaction in C3 plant photorespiration. Rubisco also catalyzes a wasteful oxygenation reaction in C3 plant photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, the first product of CO2 fixation that takes place in the mesophyll is a 4-carbon compound. In this plant, the first product of CO2 fixation that takes place in the mesophyll is a 4-carbon compound. In the bundle sheath cells, the Calvin pathway is carried out for synthesis of carbohydrate. Some tropical plants show a special type of photosynthesis called C4 pathway. In this plant, some tropical plants show a special type of photosynthesis called C4 pathway. In this plant, the first product of CO2 fixation that takes place in the mesophyll is a 4-carbon compound. In the bundle sheath cells, the Calvin pathway is carried out for the synthesis of carbohydrate. In the bundle sheath cells, the Calvin pathway is carried out for the synthesis of carbohydrates. With this, we completed the chapter. Thank you for hearing.